So it's 3:30. Should we start?
Yes, Sunat, we can start. Sir,
Okay.
next, sir.
To start.
Yeah, okay.
Start.
Okay, good afternoon, everyone. Respected Director of National Rice Research Institute, Dr. Padmini Schwein, Madam. Today's guest speaker, Professor Detlef Weigel, Managing Director, Max Planck Institute for Biology, Tübingen, Germany. All head of divisions and regional stations, scientists, students, and the dignitaries from ICAR and other institutes, a warm welcome to all of you. And a very good morning to Professor Weigel. We are very pleased to have you here. So as you know, this special talk is a part of the lecture series being organized at National Rice Research Institute for celebrating 75 years of independence of our country. We call it Azadi Ka Amrit Mahotsav, which is an initiative of the government of India to celebrate and commemorate 75 years of independence of progressive India, the glorious history of its people, its culture and achievements. The official journey of Azadi Ka Amrit Mahotsav commenced on 12 March 2021, which started a 75-week countdown to our 75th anniversary of independence, and will end post a year on 15th August 2023. The Mahotsav is dedicated to the people of India and will be celebrated as a people's festival in the spirit of people's participation. The Indian Council of Agricultural Research is undertaking various activities in this regard, and these include awareness campaigns and lectures by eminent personalities on the thematic areas for reaching out to the maximum stakeholders in different sectors of agriculture. Under the ICR umbrella, National Rice Research Institute is also organizing lecture series. Today, we are fortunate to have with us Professor Detlef Weigel, member of Royal Society of London, and an eminent scientist from the prestigious Max Planck Institute for Biology, Tübingen, to deliver this special talk. Professor Weigel needs no introduction. He has created a world of discoveries in the field of molecular biology and the evolution of adaptive traits. Very recently, Professor's team has published a very interesting paper in the journal Nature, which established that genomic regions that are functionally important for reproduction and viability are enriched with molecular features that reduce the rate of mutation in those parts of the genome. These findings challenged a long-standing principle of genetics that mutation is random throughout the genome. So now quickly coming to today's program, I would like to request Dr. P.C. Rath, Head Crop Protection Division, to welcome Professor Weigel and give a brief about the program. Rath, sir, please.
Thank you, Dr. Somanath. Welcome. Good morning, Professor Detlef Weigel and colleagues in Max Planck Institute of Biology, Tübingen, Germany, and a good afternoon, everybody in India. I take this opportunity to extend a warm welcome to Honorable Professor Detlef Weigel, Managing Director, Max Planck Institute for Biology, Tübingen, Germany, to our institute, albeit in virtual mode, on the occasion of a special talk series to celebrate 75 years of India's independence. I welcome our director, Dr. A.K. Naik, to preside over the virtual function. Hearty welcome to all the dignitaries of ICAR headquarters and other ICAR institute, head of divisions, scientists of NRI, and all other staffs. Welcome to one and all to this virtual function. As we know, our institute is celebrating Azadi Ka Amrit Mahotsav since August 2021 till August 2022 by conducting different events, and out of which special lecture series is one of the event. We have already conducted a 3 lecture series. First one on 18th August we have conducted one lecture series on the topic Agripreneurship-led extension. It is given by a profound scientist, Dr. P. Chandrasekhar, Director General, National Institute of Agriculture Extension Management, Hyderabad. The second lecture was held on 7th September 2021 on topic Genomics of Wild and Domesticated Rice by Professor Robert Henry, Professor of Innovation in Agriculture, the University of Queensland, Australia. And the third lecture was held on 7th December 2021 on CRISPR-Cas-enabled crop precision breeding and disease diagnostics by Professor Yang, Department of Plant Pathology and Environmental Microbiology, Hunk Institute of Life Science, Pennsylvania, USA. And to this series, this is the fourth lecture in the special talks to celebrate the 75 years of independence, Azadi Ka Amrita Mahotsav, going to be conducted now on 28 January 2022 on topic A Mutation is a Mutation is a Mutation by Professor Detlef Weigel, Managing Director, Max Planck Institute for Biology, Tübingen, Germany. Professor Detlef has kindly accepted our invitation to deliver this fourth special talk on this topic, a mutation is a mutation is a mutation in virtual mode. As we know, Salvador Luria and Max Delbrück made a profound discovery in 1943 that led to winning of a Nobel Prize, which was shared by Alfred Henry 26 years later. What they found was that bacterial mutation that confer resistance to a virus arise at the same rate regardless of whether the virus is present. That the generation of mutation is blind to its consequence has since become an established principle of genetics. Writing in Nature during January 12, 2022, Professor Weigel's group reported that in stark contrast to this tenet, the rate of a mutation in the model plant Arabidopsis thaliana is a lower in genomic regions that are functionally more important and in the regions where mutations are more frequently harmful. So let us listen to this and more through the special talk by Professor Detlef. Thank you.
Okay, thank you, sir. So now I request Dr. Sudhama Mandal, Principal Scientist, NRRI, to present a brief CV of Professor Weigel to the August audience. Please, sir.
Thank you, Somnath. Hello and welcome to our special talk by Professor Detlef Weigel. It's my pleasure to introduce Professor Detlef Weigel to this August virtual audience. Mutation bias reflects natural selection in Arabidopsis thaliana. It's a title of a research paper published in Nature online on January 12, 2022. Yes, I'm talking about Professor Detlef Weigel's challenging paper published in Nature 2 weeks ago. Professor Weigel's group had challenged the long-standing doctrine in evolution that mutations can arise anywhere in a genome with equal probability. Their research published in Nature shows that mutations in the genome of Arabidopsis thaliana appear to happen less frequently in important regions of the genome. Professor Weigel, a German-American scientist, is currently managing director of Max Planck Institute for Biology, Tübingen, Germany. His lab is passionately known as Weigel World. Wow, what a name. And truly, Weigel World is a world of science, a treasure trove. Professor Weigel was born on December 15, 1961. On his 60th birthday in 2021, December 2021, current and former members of Weigel Lab presented Celebration of Plant Science 2021, a pre-international online symposium with exceptional lineup of speakers in his honor. Coming to his education, Professor Weigel received his diploma MS in biology from University of Cologne, Germany, with JA Campus Ortega. He received his PhD in 1988 in genetics from Max Planck Institute for Developmental Biology and Eberhard Karls University, Tübingen, Germany. From 1989 to 1993, he was research fellow in the Division of Biology, California Institute of Technology, Pasadena, California, USA. Coming to his impressive professional experience, Professor Weigel was assistant professor from 1993 to 1999 at the Salk Institute for Biological Studies, California, and he continued there as associate professor from 1999 to 2002. From 2001 till now, he is director, Max Planck Institute for Biology, Tübingen. Until 2021, Max Planck, it was named as Max Planck Institute for Developmental Biology. The name has been changed last year. Looking towards its impressive list of his honors, awards, and accolades, I'm just bewildered to what to say and what not to say. I will be just mentioning a few of the esteemed awards he has received in his career. He is a member of the US National Academy of Sciences, the American Academy of Arts and Sciences, the German National Academy of Sciences, Leopoldina, and the Royal Society, and a recipient of several scientific awards. Most recently, the Stephen Hales Prize of the American Society of Plant Biologists, USA, and the prestigious Novo James Prize of the Novo Nordisk Foundation in 2020. His publications are of very high quality. Professor Weigel has more than 370 research publications, 323 research publications, plus 51 bioRxiv submissions, and his Google Scholar citations are at a whopping more than 100,000, with H-index 153 and I-10 index 394. He has 95 other articles, including those published in Nature, Nature Genetics, and eLife. Last year, in 2020, he published a paper in eLife that publishing in the time of COVID-19. It's a really wonderful paper in the time of COVID in this difficult situation throughout the world. He is basically among the 10 most highly cited plant scientists in the world. This is a beautiful and excellent achievement. Coming to his contributions to community and institutional services, Detlef has an extensive record of service to the scientific community. Having served on a series of editorial and advisory boards, he is a forceful advocate of open access publishing and founding deputy editor of eLife. He is also co-founder of 3 biotech startups. We are indeed delighted to have Professor Weigel for speaking to us on a topic that has taken the scientific community by storm, specifically his 2022 Nature paper that attracted our Nature podcast. Thank you. Over to Dr. Somnath.
Okay, thank you, sir. So now it's time to listen to Professor Weigel. So, sir, over to you for your presentation.
Thank you very much. Thank you, Dr. Roy. Thank you, Dr. Mandal. Thank you, Professor Svein. Thank you, Professor Rad, for being there and inviting me to speak to you in this very important year of your history. Let me share my screen. All right, so you should see a blank screen. That's on purpose, because I would like to start with a little quiz. you'll recognize, you see here 3 DNA sequences. They are slightly different, and to make it easy for you, I have highlighted where there is or is not a difference. And so the quiz goes as follows. Please look at the sequence here on top and then look at the sequence here in the center. Now, anybody who does not, I repeat, does not see a difference between the top and the center, please raise your hand. All right, I'm looking around. I don't see any hands raised. Now, please look at the sequence in the center. Look at the sequence on the bottom. Anybody who does see a difference, so I repeat, anybody who does see a difference, please raise your hand. Again, I don't see any hands raised. That means all of you have passed a test with flying colors that unfortunately the EU Court of Justice failed to pass 3.5 years ago, when they claimed that there wasn't a difference between these 2 sequences, because this is the standard genome and this is a spontaneous mutation. But they claimed there was a difference between these 2 because this mutation was derived spontaneously, whereas this mutation was engineered by genome editing. And we'll come back to this at the end. So I speak about mutation today. Thank you very much again for the wonderful introduction. And way of disclaimers, in addition to my day job, there are a few other entities from which I benefit, and they are listed here. Mutations, as you all know, are a fuel of evolution and adaptation. They are not the only fuel of evolution and adaptation. We had, for example, formation of the eukaryote by intracellular symbiosis that did not involve mutation necessarily. Also, recombination is very important. Polyploidy is very important. But mutation is really at the heart of much of evolution. Without mutation, evolution at some point obviously would stop. And when we think about mutations, we often think about phylogenetic trees. And again, you're biologists, you'll recognize the first phylogenetic tree ever drawn by Darwin himself. And that's the topic of today's presentation. I am interested, among many other things, how plants adapt to their environment, both the biotic environment, that is pathogens and microbes and other predators, and the abiotic environment. Now, we all know that we live in a time of global change, and I've brought with me here a graphic from a paper published in Nature Climate Change a number of years ago, where the authors looked at future threat of more extreme drought. And these purplish colors here, they indicate particularly high threat. And you'll notice that actually here in Europe, we are at particularly high risk of more extreme drought. Not much, for example, is going to change here in the Sahara Desert. It's already very dry, so it can't really get much drier. But in Europe, the climate has been very moderate, and that will unfortunately change in the future. So what we've been interested in, how can plants adapt to global change, specifically climate change? And traditionally, the way that scientists have thought about this, that plant will adapt by migration. So when we look in the Northern Hemisphere, the way to predict where plants will thrive in the future is you look at where a plant is living today, you draw what is called a climate envelope, and then you just look where will that same climate envelope be found in 50 or 100 years, and then you predict this will be the new range for the species. And very similar on a mountain, you predict that it will just move up the species. And here is an example from a particular paper published 20 years ago, where you see these kind of predictions where this species here, this is where it was distributed at the time, and these were the predictions for 2050 under either moderate or extreme scenario, and you see they move just north. Now, this has a few problems, this migration scenario, one of which is that plants are not known for having legs. So migration is really very different for plants. They can, of course, migrate via pollen or seeds. But another problem is that these sort of adaptation relies on populations at the edge of the range on the northern hemisphere here, either in the south or the north. And these edge populations, they are normally the least adapted and least diverse because they are really at the edge of where the species can live, and also because they're generally fragmented. So this scenario, just to repeat, has a few issues. So what we wanted to learn, what is the prospect of plants adapting in place? And the first thing that we need to know is, of course, is a plant already adapted? Now, we work with this plant here, Adaptation thayana. This is not entirely up to date anymore, but this just shows that the plant is distributed very widely. Long was thought that its origin was in Europe and Western Asia. We now know that the origin was actually in Africa, and then it was later introduced to North America, to South America, to Australia, and so on and so forth. So it occurs pretty much around the globe, and it lives in very different environments. This is near Tübingen, an abandoned train track. This is an oak forest in Spain. This is not a staged picture. This is an actual plant growing on the banks of the Baltic Sea, an abandoned cloister in Ireland, and here in Patagonia. So you get the idea. This plant lives in very different places, must be adapted to very different places. And so this is something that Moy Exposito Alonso, who is now a PI at the Carnegie Institution, asked a number of years ago. How well are these plants actually adapted? He drew on the 1001 Genomes project, where we have completely sequenced the genomes of many different strains of Adaptation thayana. He selected about 500 of these strains, and then he grew them either in Tübingen, where they're naturally high precipitation in these rainout shelters, or here near Madrid, where there's naturally low precipitation. And so they experienced the normal climate in these two places, but the rainfall regime was manipulated. And you can see right away when there's mild drought, plants do not nearly as well as they do when there is simulated rain. So Moy imaged 24,000 pots over 20 time points. He counted survival of the plants. He counted the seeds for both individuals. So some pots had only one individual, or for populations, other pots had 20 seeds. So looking at survival, looking at reproduction, using AI type of methods to estimate how many seeds each of these plants produced. And this is what he was able to do then, directly observing lifetime fitness. And you see right away when there's drought, plants don't do nearly as well when there is rain, not a big surprise. Drought is not nearly as lethal in Tübingen as it is in Madrid. So you see many plants produce no seeds at all when there's drought in Madrid. And that, of course, comes because in general, the climate and the weather in Tübingen is less harsh than it is in Madrid. And then there's a benefit growing under high density. Generally, plants do better than growing under low density. So because these genomes were all sequenced, Moy could then make use of these millions of known alleles and ask for every allele in the genome whether it was associated with increased or with decreased fitness. And so this was really the first time anybody was able to do this really across the entire genome. And what it was able to show was indeed there's rampant evidence for local adaptation. I should say here, of course, this has been known before that there is a home advantage often, but this is typically was based on reciprocal transplant just among two strains or very few strains where you would transplant strains from Madrid to Tübingen and vice versa. Whereas here we take strains from all over the range. And so what is shown here is where the alleles come from based on annual precipitation from the home of these alleles. I'll say a little bit later how we determine the home of the different alleles, and then the precipitation seasonality, that is how even is precipitation throughout the year. And so Madrid, for example, comes out here, Tübingen comes out here in this two-dimensional map, and then each of these millions of alleles is colored whether they are positively selected in Madrid or selected against in Madrid. And you can see that alleles that come from places that are similar to that of Madrid, they are much more likely to be positively selected in Madrid than they are alleles that come from places where the climate is very different from Madrid. They are much more likely to be selected again. So a lot of red here and a lot of green over here. And so from that, you can predict what will happen in the future. So today there is already high selection intensity here around the Mediterranean. And then when you look at how the climate will change in the future, we'll see there will be this northward wave of selection that, of course, all makes a lot of sense. So an important question then is how quickly can plants actually adapt? So to address this question, again, Moy here did another experiment. He asked how easily can plants survive extreme drought. So he again took accessions from the 1001 Genomes project. This was a little bit of a smaller experiment. And then he grew these plants in the greenhouse. And the important thing is he watered them for a while and then he withdrew water. There was a little accident here. One of the undergraduates hadn't gotten the memo and thought the plants looked really sad and gave them a little bit of water off schedule, if you will. But you see, in general, soil water greatly reduced in the time course of the experiment. And so this is what the plants look like when you observe them over time. You see they disappear. It's actually the plants disappear. It's just we here only show the green pixels, and as the plants dry up, there are fewer and fewer green pixels. So from these images, these green pixels then can estimate the survival of the plant. So this is tracks for individual plants. And you see here how the green pixels decay. And based on this, you can come up with, Moy came up with a drought survival index, which is nothing else, just the quadratic parameter that describes the decay in green pixels. And here you see, for example, a rather drought-resistant accession at the upper end, and here you see a very drought-sensitive accession at the lower end. And so then this quadratic parameter is our drought survival index. Now, we know where all these accessions come from. We know where other accessions that we didn't include in the experiment grow. So we can perform environmental niche modeling using random forest to estimate an accession, even if we haven't seen it. If it came, for example, from this place, how well would it survive? Would it have survived in our experiment? Well, you see here in Central Europe, indeed, most of the accessions wouldn't do very well in our experiment. Around the Mediterranean, they do a lot better. Not too surprising because that's already pretty dry. A surprise was that accessions that come from Sweden, for example, that they would do quite well as well under drought. That was surprising, but then we thought about this and we realized plants here overwinter under snow. And under snow, there is no water because all the water is in snow. It actually constitutes a drought environment. So again, this makes a lot of sense. Now, having done this, we can produce GWAS and climate maps for both good and bad alleles, meaning good alleles, those that promote survival under drought, and those that are bad under drought. So we basically first perform GWAS. We nominate genes and alleles that are predictive of performance. And then we ask, where are the accessions that have either the good allele or they have the bad allele? And then we can, again, draw a distribution, and we estimate where is the center of the distribution. So for the purple allele, for example, the center of distribution would be somewhere here. And for the yellow allele, it would be offset somewhere over here. So we can do that for one allele. We can do that for all the alleles that we have. And then we get a local density of the alleles that either promote survival under drought or that don't. And what you see here, that in the center of Europe, the density of alleles that promote survival under drought is very low, whereas the density of such alleles is high around the Mediterranean and also relatively higher here in Scandinavia. And these are just examples of actual accessions that come from these different places. So having this all in hand now, we can make predictions about the future. We ask, what are the alleles that plants have today? What is the climate today? And then we can ask, so in 2070, for example, what is the climate then? And how many alleles will they need to survive? How many more alleles will they need, or fewer alleles than they have right now? And from that, we can calculate an adaptation deficit. So if they need a certain number of more alleles, we can ask how many alleles are currently there. And if there are fewer than are actually required, then there is an adaptation deficit. And again, here, the surprising result that actually here in the center of Europe, accessions are most at risk, populations are most at risk, whereas they are relatively fine around the Mediterranean, they are relatively fine in Scandinavia. So this is work that we did with a wild plant, Arabidopsis thayana, obviously not a particularly important plant, but I think it gives you an idea, the sort of approaches that you can use to make predictions about the ability of plants to adapt in the future. Now, as I said, it looks like it's relatively grim for plants that grow here in the center of Europe. So this, of course, begs the obvious question: can plants adapt from new mutations, and are new mutations limiting? So that is the backdrop for our work on mutation rate and spectrum. Now, you all remember from college that that there are many different causes for mutations, replication errors, spontaneous deamination of methylated cytosines, for example, reactive oxygen species, UV activation of transposons, toxins here such as aflatoxin from fungi. So many different ways in which DNA can be damaged, even if you use sunscreen, and plants typically use sunscreen in terms of in the form of pigments. Now, fortunately, most DNA damage is very effectively repaired. Many different mechanisms: direct reversal of damage, single strand damage repaired, double strand damage repaired. And this really works very well in all organisms. And that's really good because that prevents us from having disease. But of course, repair mechanisms are not perfect. And the very few instances where repair doesn't happen, mutations arise. Now, thinking back to Darwin, when he came up with the theory of evolution by natural selection, he didn't actually say too much about mutations and how random they were or not, but generally now we associate Darwin with this idea that mutations are random with respect to their benefit to the organism. In the introduction, the famous Luria-Delbruck experiment was managed, where it was found that mutations that make bacteria resistant to a phage are already present in the population before the bacteria have ever seen the phage. So again, there has been this prevailing idea that mutations are entirely random with respect to their outcome. And of course, that in a way makes sense. In organism can not know beforehand whether a mutation will be beneficial or not, because it has to first try out what that mutation will actually do. So this central tenet of Darwinism, or more correctly, neo-Darwinism, is that mutation is random. And then what we observe, that we call polymorphism, is actually the result of natural selection, is a sieve that removes mutation that harm the organism. That, of course, remains true despite our work. But I want to point out that random mutation and natural selection are, of course, entirely sufficient to explain the pattern that we observe. But even if mutation was not entirely random, natural selection is still operating. So natural selection is really what's essential. It can work with random mutation, but it's not an absolute precondition that mutation has to be entirely random. And we already knew for a long time that with respect to, for example, DNA, local DNA composition, DNA modification, and so on and so forth, there is variation in the rate of mutation along the genome. So for many reasons, knowing the natural mutation spectrum and rate is something that is really important when you want to understand evolution. And so scientists have for a long time thought about how to measure mutations. And for that reason, this idea of mutation accumulation lines was developed actually about 100 years ago by Muller. And this is from a review by Halligan-Keatley that lays out the principle. You start with an ancestral line, you split it up into different sublineages, and then you propagate these sublineages in cell-phase plants by single seed descent, or in organisms where you need different males and females, you would just do brother sister matings. And so when new mutations arise, they would become either homozygous, or a new mutation would get lost because the wild type allele becomes homozygous again. So new mutations arise, 50% of mutations are lost upon selfing. You do that for a certain number of generations, and at the end, you look at these lines and try to estimate how many mutations arose. Now, as I mentioned, this idea is actually old. So Muller first came up with this. It's really quite interesting, this paper in 1928. He was a little bit worried that mutation, although he knew it must exist, was something that was not really observable with scientific tools. Although in this really beautiful paper, he solved this problem and actually introduced mutation accumulation lines and showed that he could actually indirectly observe that lethal effect mutations had arisen in the Drosophila strains that he was maintaining. So can one measure mutation rates directly? Fast forward almost 100 years. When Arabidopsis thaliana became more and more popular, different groups of scientists started to generate mutation accumulation lines also in Arabidopsis thaliana. And one can indirectly estimate mutations even without looking at genomes by looking at fitness and then making back of the envelope calculations, if you will, how many deleterious mutations are present in these lines to explain the changes in fitness that are observed. Now, fortunately, when next generation sequencing was introduced, we remembered that Ruth Shaw, colleague in Minnesota, had generated a set of mutation accumulation line that was in the Columbia 0 reference background. So it was in the background from which the very first Arabidopsis thaliana genome sequence had generated. So that made it particularly straightforward to sequence these lines with these very early instruments for next generation sequencing and count mutations in these lines. And so this is an image, a graph from the paper published in 2010 with Stefan Ossowski as lead author. And this is a result from sequencing 5 lines. Each of these lines had about 20 mutations. So you can see this is quite a challenge, 20 mutations among 100 million base pairs. So 1 out of 5 million. So this is truly finding a needle in the haystack. And again, became only possible with these new methods of sequencing where we can sequence every genome many, many times so that sequencing error essentially disappears and we are left with the mutations. And so what we could estimate that there is about 1 new mutation per haploid genome and generation. So give or take 10 to the minus 8 per base per generation. That comes down to about 0.2 non-synonymous mutations per haploid genome and generation. Now, that's a very useful number, and it has been used widely in all kinds of evolutionary and population genetic analyses. A question is, of course, this was done with plants that were raised in the lab, and is this really is this the real mutation rate, if you will? So to better estimate what it looks like in nature, we could have taken some seeds, put them outside, leave them there, come back after 10 years or so, and then sequence those. Would have been one option, but then we realized that apparently there was a natural mutation accumulation experiment that sort of had been conducted in North America, of course, somewhat unknowingly. And this came from our looking at the genetics of North American Arabidopsis and different from Europe. We found that there was one lineage, this purple lineage here, that was very widely distributed. So in Europe, you would have a single lineage only at a single place. But this indicated that perhaps there was a lineage that was introduced to North America early on when Europeans colonized North America and spread widely and behaved as a natural mutation accumulation experiment. So, and that of course would be ideal to try to estimate mutation rate. And the idea is we would have lines that have been collected relatively recently. They have been collected at somewhat different times, say in 1990 and in 2010, so 20 years difference. And then this lineage here should have accumulated a few more mutations relative to this one. Now, if this really goes back, as indicated here, a few hundred years, then 20 years difference here at the tip is not very much. So what would be a lot better if we somehow could also sample earlier examples of this lineage. So for example, here I have one where evolution would have stopped earlier and therefore fewer mutations had been accumulated, and this difference would be much larger. So here what we need is a time machine. Unfortunately, time machines do not exist yet, at least not in this form, but there's a very different type of time machine. And I really have to credit my postdoc mentor, Elliot Meyerowitz here, was really his idea. When I presented our mutation accumulation line work, he said, well, did you know that Arabidopsis thaliana is really quite abundant in herbarium samples? So we got our hands on herbarium samples. We used a proper technique to extract DNA from these samples. And we ended up with this collection where we had samples from essentially every decade since the 1860s. And then we also added modern genomes from this lineage here that we call HPG1. That was work that was led by Claude Becker in collaboration with Anand Bobano. And that then allowed us to calculate the substitution rate. Here you see a phylogenetic network of these modern and herbarium genomes. It's a network. You don't see any reticulation because it actually behaves like a clone, as you'd expect. And you see that on average, the branches here for the blue samples are shorter than for the green samples, consistent with this idea that evolution stopped earlier for these blue samples than the green samples. And again, we can triangulate the substitution rate, and it comes out to something 2 to 5 per times 10 to the minus 9 per base per year. So quite similar to what we see in the lab. We don't know exactly what the generation time is outdoors, but we estimate that the generation time would at least partially account for this difference in substitution rate if the generation time is a little bit under a year in the wild. All right, so we measure this rate. That's a number. It's an interesting number, very useful. But is there anything else interesting in the mutation accumulation line data? So over the time, sequencing technology became better and better. We continued to sequence mutation accumulation lines. And this is from a paper, Maolun Weng was the first author, published in Genetics in 2018. We confirmed a pattern where we had seen some indication already in that 2010 paper that mutations were less frequent on chromosome arms than near the centromere, and that was true for all 5 chromosomes of Arabidopsis thayana. So there's something going on. So as an Arabidopsis biologist, when you see this difference between chromosome arm and centromere, what you immediately think of are differences, for example, in chromatin accessibility, differences in cytosine methylation, CG methylation, CG methylation being much more common around the centromere, chromatin being much less accessible around the centromere. But there are, of course, many other things, many other features of the genome that vary along the chromosome. So Gray Monroe, postdoc in the lab, now an assistant professor at UC Davis, joined the lab and wanted to revisit this, whether there were more interesting patterns. But the first thing that he wanted to convince himself of was that really there was very little selection in the mutation accumulation lines, that the mutations that we saw in the mutation accumulation lines that we observed were a true reflection of actual mutation, and that selection wasn't blurring the picture. So when you look at the ratio of stop mutation to synonymous mutations, individual genes, then you see this ratio is very low in the 1001 genomes collection. So these natural strains of Arabidopsis thayana is very similar in the mutation accumulation lines compared to a null model. And similar, when we look at the ratio of non-synonymous to synonymous mutation, non-synonymous mutation clearly depleted in the natural lines, but there is little difference between the mutation accumulation line and this null model here. So this is data. These are data from over 100 lines. This shows indels and single nucleotide variants along the genome in different bins along these 5 chromosomes. And that's the work that was mentioned earlier that was published just a couple of weeks ago, led by Gray Monroe, now at UC Davis, and inspired on the chromatin side by work from Tanvi Srikant, who just graduated and is now a postdoc at ETH with Kirsten Bomblies. So what Gray wanted to look at were there features along the genome that might be predictive of mutation rate. So he took somatic and germline mutations, almost 10,000, and then he gathered data on gene expression, on GC content, cytosine methylation, and so on and so forth. And he built models where these sort of features would predict the local mutation rate. And you can see here that different features have more or less predictive power. So, for example, GC content is strongly anti-correlated with single nucleotide variants and also indels, whereas chromatin accessibility is positively correlated with both of these. Now, having this model, knowing which features matter, then what you can do is you can annotate the entire genome. And I should mention that he got very similar results whether or not he excluded coding regions for training. So you can then annotate the entire genome and you can look at mutation probability. And so here, for example, mutation probability around genes, you see that in front of transcription start sites, that is outside genes, mutation probability is quite high, whereas in genes it's low. And then the opposite pattern here, when you encounter transcription termination site coming from this side, where you see inside the gene, it's quite low. And then as you leave the gene, it becomes elevated. And that you have these dips here. This just comes from the back. As you move away from the transcription start site, you tend to encounter new genes again. And similarly, as you move away from the transcription termination side, you tend to move into genes again. And we didn't break it down by whether or not there were adjacent genes. Now, these are really intriguing patterns, but can we validate this in some way? So Gray really went to great lengths. He looked at germline mutations in a completely independent set of mutation accumulation lines. He looked at somatic mutation lines and mutations in these lines. He looked at somatic mutations in an independent data set published by others. And he also looked in these North American lines. And always he sees the same pattern where around transcription start sites outside, mutations are much higher than here inside genes, and the opposite pattern around transcription termination sites. Turns out natural polymorphisms behave similarly, and these are polymorphisms in 1001 genomes. And so, of course, this could be much due to selection, but more importantly, Tajima's D also shows a related pattern, and that it's less negative inside genes here and here, tells you that these mutations are rarer and rarer and younger. So clearly consistent with this idea that there are fewer mutations inside genes. Even more interesting, when he looks along genes, breaks it down by genes with different exons, see that the first and the last exon are always the one that have the highest mutation rate. And again, that's confirmed in the polymorphism data from the 1001 Genomes Project and also in Tajima's D pattern. And it's also seen in a completely different species, Populus trichocarpa. Again, these are natural polymorphisms, and they show this really intriguing pattern where the first and the last exons are one always that have the highest rate of mutation. So having these data then go further, and Gray said, let's see whether we can maybe do something even more interesting. So let's break down gene classes according to mutation probability. So he assigned a mutation probability to every gene in the genome. And then he asked, does that correlate with anything? So he asked, for example, when genes are annotated as either being essential or non-essential, what does their mutation probability look like? And it turns out that non-essential genes, they have a higher mutation probability than the essential genes. Also, when they have a mutant phenotypes and they are annotated as either essential or having morphological defects, biochemical defects, or conditional defects, again, you see that the genes with conditional mutant effects have the highest mutation probability compared to the essential genes. And this is confirmed with direct observation in coding sequences. Here, you, of course, you can say in coding sequences that includes potential knockout mutations. Obviously, many mutations will not affect the function of the gene, but still you could say much of this difference, although this is a really, really large difference, almost a twofold difference, you could say at least some of this could be due to selection, but we also see it in introns where obviously most mutations do not have an effect. So this this observation environmental, when there's an environmental defect only, much higher mutation rate than when genes are essential. And then finally, he looked at when we break it down, say in the bottom decile, the top decile, and you see it's really quite a range of mutation probability. Are these actually different genes by their function? And indeed, he finds that the genes in the bottom decile of mutation probability related to processes that are universal in cells, such as translation and splicing, whereas those in the top decile, they are much more likely to be related to response or regulation. So that, of course, sounds amazing that genes that are particularly important for the organism or for very widespread function in the organism mutate less than those that are not. That begs the question, could there be a plausible mechanism? Well, I already had said that the model was built on chromatin marks, and indeed what we find is that chromatin marks are highly correlated with evolutionary constraints. So he has one such mark, histone 3, lysine 4 monomethylation. And here for genes in 50 different deciles, you see the anti-correlation with the number of synonymous polymorphism per base pair, or a divergence between Areopsis thayana and Lyrata, synonymous and non-synonymous per base pair, the ratios of these two, and then also gene expression. So how stable gene expression is either in response to the environment or in different genotypes. And you see that when H3K4 methylation is high, a high substitution rate, high variation in gene expression. And then when we look at the observed de novo coding sequence mutation, we see this opposite pattern. So sorry, so the polymorphisms, sorry, we see the opposite pattern here with the de novo mutations. So, and if de novo mutations are few, then of course this is the inverse. Then the number here is higher. All right. So, what is then the bottom line? So what we suggest, looking at the genes, upstream, downstream, and the gene body, well, we posit that they are epigenome regulators that lead to gene bodies being differently marked with chromatin marks, and therefore attracting DNA protection and or repair machinery more effectively. And it turns out there's plenty of literature that actually shows an intimate connection between chromatin marks and DNA repair. And so similarly, this is actually the main observation that we have made, is that essential genes, such as housekeeping genes, as a class, are marked in a different way than non-essential genes. And that allows these genes here as a group to more efficiently attract DNA protection and repair. And that's the reason when we first put this on bioRxiv that I tweeted that I thought probably the most provocative study ever from my lab. So this has really changed how I look at the world around me. Here is a stock picture of a wheat field. This is a large wheat field, obviously much larger than 1 hectare. And these are all different plants. So I told you mutation rate is approximately 10 to the -8 per base per generation. A wheat genome is 20 gigabases, so we can estimate about 200 mutations per generation. 1 gram of wheat is about 20 grains. In Germany, in a good year, a hectare of wheat will produce about 10 tons. That's 2 times 10 to the 8 grains per hectare. This would actually be 40 billion mutations in the harvest from a single hectare. And so the genome only has 20 billion base pairs. So on average, every base pair in the genome mutated twice with variation along the chromosome. But the important thing is these are all mutant plants, and they're all different from each other because they have different mutations. And of course, genome editing is nothing else than targeted mutagenesis as opposed to spontaneous mutagenesis, and for which very satisfyingly Emmanuel Charpentier from Max Planck and Jennifer Doudna from UC Berkeley were awarded Nobel Prize in Chemistry a year and a half ago. And I should point out that even the Intergovernmental Panel on Climate Change has pointed to genome editing as a tool that might be useful in combating the consequences of climate change. Unfortunately, not everybody else thinks the same. This is a protest against these new gene technologies, new GMOs in France. And it is a bit ironic that our own work here, this was the reference to the preprint of the Monroe et al. Paper, has actually been twisted by the opponents of genome editing, where they essentially claim that we had shown that there are genes where mutations are very unlikely to occur naturally. And of course, you have seen the data. What we've shown is that mutation rate is reduced by a factor of 2. I would not say that is very unlikely. And of course, these genes still mutate. But that's also why it's very important to speak out in public. I'm really quite proud here, for example, commentary in Der Spiegel, leading German weekly, that pointed out that I'm one of the few well-known scientists in Germany who insert themselves into these sort of public discussions, but I'm really encouraging all of you to do the same. So coming back to where we started, a mutation is a mutation is a mutation. I hope I've given you a little bit insight into how common actually mutations are in the genome. That also really puts genome editing in perspective. If we change one place in the genome, even if there are a couple of off-target effects, compared to what spontaneously happens in a genome in every generation, it's really a drop in the bucket, if you will. And we have actually shown directly that performing genome editing, if you don't know where the edit is, it does not measurably increase the genome-wide mutation rate. So after you have genome edited the plant and you count the mutations that you have in the plant, if you don't know what the genome edit was supposed to target, you cannot distinguish that plant from the plant that has not been subjected to genome editing. With that, I'd like to thank my lab, and especially like to thank Max Planck for allowing me to do all this work. And I thank you for listening, and I'd be very, very happy to answer questions. Thank you very much.
Thank you, Professor. That's a fascinating presentation. And applaud to you and your team for such a nice work. So now there is about 10 to 15 minutes for discussion. And I request the audience to raise your hand or post your questions in the chat box so that we can interact with Professor.
Thank you, Lambodar, for the nice words. Maybe I can ask, I can start with a question to you all. I would be really curious to know what public opinion, if any, is on genome editing in India, and at your institute. Does genome editing play an important role? Do you think there is a role for genome editing in your efforts?
Dr. Kutub, you can respond to Professor query.
Thank you very much, Dr. Detlef. So wonderful, because just a few weeks ago, I read your Nature paper and listening to you about that same paper. So yeah, genome editing, you know, we in our country, the funding agencies and from the policymakers, they are insisting on taking up the project on genome editing because it has a big role to play in Indian agriculture. If you consider regarding developing climate smart varieties of disease and pest resistant varieties and all these things. So there are a lot of projects going on all over India, and even our Department of Biotechnology, Government of India, they have drafted a draft regulatory rules of genome edited crops, and that has a lot of positive sign in it. So if government goes with the proposal, then the SDN1 and SDN2 type of mutations created by genome editing would be allowed without going through the conventional GMO regulation. So that's a very positive point for all of us who are working on this line. But I would also like to know from your view, so what the European Union is thinking, because you know, we have heard about the negative judgment I think 1 year ago. So is there any positive means U-turn regarding the regulations in European Union?
Yeah, maybe I can speak to that a little bit. So there was this judgment by the EU Court of Justice in July 2018. So that's 3.5 years ago now. It was brought by French farmers. The ruling was actually somewhat of a technical ruling. I have to step back a little bit. So this is going to take a couple of minutes to explain this, but this background is really quite important. So it goes back to the original EU directive on the release of GMOs, the release into the environment of GMOs from over 20 years ago. And So the interesting thing is that it defines as genetic engineering any genetic modification. So mutagenesis is actually defined as genetic modification. The directive goes on to say that methods that have been used for many decades, such as mutagenesis, they have not been found to be particularly dangerous, and therefore they are exempt from the directive. So one way to interpret this is that the directive actually says GM, by definition, does not have to be dangerous. There are forms of GM that are not more dangerous than what happens naturally. But there are certain forms of GM where at the time there was little experience. And they say, because we have little experience, we have to regulate them. And I think that is understandable. The directive was published in 2001. The large-scale release of genetically modified plants was, it began in early 90s, but really large scale was only from 96 on with Monsanto's herbicide-resistant crops. So really at the time when this was drafted, it's true, there was really limited experience with this. What is in hindsight odd that the directive didn't say, if say in 10 years and 20 years, we have experience with this method, we have found it to be safe, then we should change the directive, the law, adopt the law. It didn't say that. It's in that case, in that way, it's actually a bad law because it didn't have any provision for changing it in future. And so this is what the definition of the EU Court of Justice hinges on, and says these new ways of genome editing, we do not have experience with them. And the directive says if we don't have experience with them, they must be regulated. The irony is, of course, that there isn't a provision to really change this once there is experience. So this is really what it hinges on. What the Commission in the meantime has done, it has commissioned an assessment. The assessment came to the conclusion that the directive is outdated and it must be changed. There's a lot of lobbying going on. My colleagues and I are involved in many different levels to try to change the law. What the outcome of this is difficult to predict. What I'm a little bit afraid is that at the European level, the European Union will say, genome editing, we only need light touch regulation, but every country can have stricter rules if they wish. So I'm really afraid that there's going to be, really a chaos. The UK is leading, which I'm sure you have seen. In the UK, it's quite clear that almost certainly there will be much more light touch regulation of genome edits, maybe not quite as light touch as in the United States, but more light touch. And then also in the EU itself, Sweden has already said, essentially we don't have to regulate genome edits. Belgium has also said, very little regulation. In Spain, that's sort of unknown. Spain is the only place in the EU where traditional GM plants have been quite widely grown. So in Spain, that's also not a big deal. But then there are other countries which are very much on the other extreme, beginning with Austria, but Germany not much behind. Our new government, the good news is that one of the parties in the government is the only party in parliament that is actually pro-GM. It's unfortunately not predictable what that government will do. The Minister of Agriculture and the Minister of the Environment, they are both from the Greens, and they are adamantly opposed to genome editing. So I think it's sort of early, but that's also why I really like to speak about these spontaneous mutations in public a lot, to just bring home this idea that genomes change very, very rapidly, that natural mutations are really rampant. And I think that's also important to think about this directive from 20 years ago. We basically had no idea how variable genomes are. So one thing that in the when I talk to the public, I often present, you can take 2 lines of maize. They look the same to me, but when you look at the genes, out of 30,000 genes or so, the one line has 2,000 genes that the other one doesn't have, and vice versa. Or that there are many species of plants that are naturally transgenic. This is all knowledge that we didn't have. It's very clear against this background that this law is completely out of date.
Yeah, thank you for your insight. So, I read an article today on bioRxiv that whitefly, they acquired 24 genes from the plant they fed on. So, so this kind of horizontal gene transfer from a plant to insect, and those are all convincing story we have, but still we lack in convincing those opponents. What do you think? What kind of measures we should take?
Well, people often say it's a matter of education. I don't think it's a matter of education. I think it's a matter of how you appeal to people. And so, since I brought up the Greens, one of their hero is Vandana Shiva, who, I'm sure is very well known to all of you. And once you look into, it's not a matter of education. I think you have to tell the public what Vandana Shiva really, stands for. That she has very questionable views on vaccination and so on and so forth. So I don't think it's necessarily educating in terms of the technology and the science, but appealing to people on a different level. And especially with global change, with climate change, I hope that we have a chance there. There are quite a few questions in the chat, so maybe I could try to address a few of those. So the first question was from Joydeep was, why more mutations than promoter than the coding sequence? And that has to do with these epigenetic marks. There are certain epigenetic marks that you find in the coding sequence, but not outside the coding sequence. And these epigenetic marks are associated with lower mutation rate. Selection bias towards genomic homeostasis. Well, I mean, there's selection bias in the sense that, of course, selection is much stronger on essential genes than non-essential genes. A question from Ramavta, a question from Gyanendra. How do you relate genome editing versus physical or chemical mutation in terms of efficiency and cost-benefit ratio of complex traits? Well, I think in when you do complex trait mapping and say you have many candidates, I think genome editing really also has real potential there. There are now methods to at the same time introduce a large number of guide RNAs. And let's say you have 50 candidate genes, you can introduce, say, 50 guide RNAs into the genome and then generate a segregating population where individuals have random collections of these 50 genes edited and then screen directly for the ones that have the trait of interest. So I think genome editing, also when you have to make many edits, can be helpful. A question from Professor Swine, I think. So thank you, Professor Swine, for your thanks. A question from Sujoy. How likely how similar are the mutation rates in other organisms? So it has not been measured in very many plants, but indirect estimates are quite similar. And then what's the contribution of epigenetic mechanism in adaptation to environmental change or evolution? So I would say yes, for sure there is contribution. What is the amount? I can't put my finger directly into this, but I would say it's probably a minor contribution. It's really quite interesting to see that many breeding companies about 10, 15 years ago, they were super excited about epigenetics as a tool and invested millions and millions, and they have all disbanded their programs. Parameswaran also has raised a hand. So maybe you can go ahead and ask me.
Permission, please interact.
Sir, this is really a nice presentation. I had a very informative talk from you. So I'm a little bit surprised. Arabidopsis, I heard that it was originated from Africa, but the distribution is not there in Africa, but it is highly distributed in Europe and North America. May I know the reason for this?
Well, that's because most Arabidopsis biologists live in Europe, but there has been, as I said, the map was a little bit out of date. So there has been a lot of collection by Padej Flood, who was a postdoc with my colleague, Angela Hancock at the Max Planck Institute in Cologne, and they have greatly expanded the collections in Africa. So apparently quite easy to find in Northern Africa, but has been found, for example, also on Kilimanjaro, and so on in Southern Africa. So initially there were just mostly herbarium specimens, but now also a lot of live specimens. But it's just a sampling artifact, really. So there's also Arabidopsis in India, certainly in the foothills of the Himalayas, and I suspect it's also quite common, but just has not been sampled as densely.
So may I ask another one of few questions, sir?
Sure, please.
Yeah, okay. So why there is some drastic change in mutational probability score from UTR to TSS start site or last exon to translation stop site? Why there is some drastic changes are there in that probability score?
Yeah, it really reflects some of these marks. So also there's a type of cytosine methylation called gene body methylation that's also very different from gene bodies and outside genes. So when you look at this, actually in the paper is one of the first figures, it shows these the distribution of these chromatin marks. And when you look at them, the paper is open access, so anybody can look at it. When you look at these chromatin profiles relative to transcription start and transcription stop, you see that they are marks that are really specific to the gene bodies, and then often they taper off as you approach the end of the gene. And so of course teleologically it also sort of makes sense, mutation rates higher in the terminal exons, the first and the last exons, because if you have a mutation in the first exon, you can perhaps get reinitiation, or you have a mutation in the last exon, you lop off a little bit of the protein that often doesn't have much of an effect. But it's really related all to these chromatin marks.
Okay, so in that case, I was also interested in seeing one data that the mutation probability score in natural populations as well as the population created in your lab for the study, they were same for genes. Mutation probability score for the genes are same in natural population as well as your population in the study. So does that mean all the mutations are always there is some bias in natural population also, or it's only laboratory study indicates this findings?
Yeah, so in the natural data, the data set is not as large, so we see this pattern around genes. There we wouldn't have enough data to actually build a model, but because the pattern around genes is very similar, we assume that there's also the same difference between essential and non-essential genes. So again, that's actually important to remember that these inferences about essential genes, this is based on the modeling. The mutations that kill the organisms, we could not. Somatic mutations, we could observe them, but germline mutations, if they kill the organism, we couldn't observe them either. Although, of course, there are not only even essential genes, there are many mutations that don't do anything to the gene. But again, really important to keep that into mind, the modeling aspect of the work.
So my last question would be, if there is very minimum epigenetic mark, say in bacteria or from lower group of organism, then in that case, can we say mutation is always random, but only in organism which is having high epigenetic marks, in that case, the mutation is biased? Is my statement right? So just I want to have your opinion on it.
Well, actually, the first observation, this difference between essential and non-essential gene, was actually made in E. coli. It was a little bit more indirect because it wasn't really mutation accumulation lines, but it was comparing closely related E. coli strains. But a really beautiful paper by Inigo Martin-Correia, published in 2012 together with Nicholas Luscombe in Nature, where they reported this effect. So this was highly controversial. And was highly controversial for two reasons. A, they did not know what could be a mechanism for this. We still don't know what could be the mechanism, although we today know that there is also a form of chromatin in bacteria. I do not know how much variation there is in chromatin along the chromosome in bacteria, but they have proteins that associate with DNA. But one weakness was they didn't have a mechanism. So our advance is really not the discovery essential versus non-essential gene, but really proposing a plausible mechanism. And also there was a lot of pushback, there was a lot of theoretical work that has shown very clearly that reduced mutation rates in individual genes could never evolve. It's just too rare. The way you can think about this, if I want to sell you something and I say, I give you a 10% discount, but it's something that only costs 1 cent, or let's say cost only 1 rupee, and you only need to buy one of these, then you say, well, 10%, if it's 90% of a rupee is still a rupee, so why should I buy it from you? But if you buy many, many of these, hundreds of thousands of millions or so, and say the total price is 1 lakh, and then it's a 10% discount, you will probably buy from me. And so this is the same with these genes. One individual gene, it cannot evolve, but all genes together, then the small difference all of a sudden matters, and it can actually evolve. And that's also something that we show in the paper with the proper math.
Thank you, sir. Thanks a lot for answering all my queries. Thank you. Thanks a lot.
Okay, sir, we have some few more queries. So Dr. Chattopadhyay, you can interact.
So I'm just reading this. So salinity tolerant genes, very much influenced by environment. Yes, I mean, that would be, so I don't have that handy here, the data, but I expect that genes such as HKT1 would have higher mutation, would have a higher mutation load. Yeah, so and then so the good and bad that I used, this was more about the alleles, whether the good allele, I meant an allele that's good for drought adaptation. So that really had nothing to do with good or bad mutations. So I'm not saying that all mutations in essential genes are bad and all mutations in non-essential genes are good. That's clearly not the case. It's just you can also show in essential genes, when you of course there will be beneficial mutations as well, but if you reduce the number of deleterious mutations by reducing mutation load in essential genes, that overall has a net positive effect.
Okay, Dr. Shopna.
Yeah, and then with the mutations, randomness, so it's still important. So to even Darwin, he didn't, he was pretty agnostic in terms of randomness. So you have to keep in mind that this is just a reduction is actually, so genes mutate about 50% less than what's outside genes, and essential genes about 1/3 less than non-essential genes. So, obviously you still really, really need natural selection. So the effects for an evolutionary biologist are large, 50% or 1/3 is a large effect for an evolutionary biologist, but it does not mean that you don't need natural selection anymore. So it's really no contradiction to the prevailing dogma. So it turns out that, again, what I tried to say in the beginning, natural selection can work perfectly with entirely random mutation. But it also still works when mutation is non-random. And I think this has been sort of the confusion that because as humans, we like things to be simple. And of course, the simplest scenarios, mutation is completely random. Natural selection is the only thing that needs to work. And now we find it's a little bit more complicated.
Okay, Professor, thank you. One last question from Dr. Shopna of Lucknow. She said, will the source of mutation have an effect on the differential rates of mutation observed across the genome?
Well, the source, we haven't looked at that. So I would think, so mutations have to be repaired. If what's behind it is really DNA repair, then I think the source of mutation will have less of an effect, although, you know, certain chemical or physical mutagens, of course, they have preferences for different composition of DNA, basically. So that will have an effect. I think is most unlikely what it means for genome editing, that it will be a little bit more difficult to make genome edits in essential genes than non-essential genes. But again, we're talking about, you know, a factor of 1/3. So which, when you work in the lab, is probably not really important.
Okay, then thank you. So we should stop here. And thank you very much for your active interaction with the participants. So now we go ahead with the program schedule, and it's time for the director's remark. I want to inform that our director, Madam Dr. Swain, is not feeling well today. So I request Dr. Nayak, Head Crop Production Division, to deliver the remarks. Nayak, sir, please.
Thank you. Good afternoon to everybody. Thank you, Professor Weigel, for your scintillating talk on mutation. A mutation is a mutation, a mutation. An interesting topic that we are waiting for. In fact, your presentation set the ball rolling for a debate and a discussion in the time when this country is in the process of framing the regulation for these guidelines for genome editing. And your talk with respect to this mutation, evolution, adaptation, and climate change, it is really educative. But we know, as a biologist, we know the 6th extinction is going on. How these tools, genome editing tools, and the science of mutations, with all the things which you have presented, whether the randomness and the targeted mutation and the measurability and the target gene, good mutation, bad mutations, all those science of mutation, how that can help us in future to adapt better to the climate change. That is the major goal of our Earth planet. And UNFCCC also recognize the genome editing is one of the tools that helps for fighting the climate change. So I think your presentation will help the natural biologist to understand the evolution and the mutation vis-a-vis the climate change. Your presentation with respect to GWAS versus the climate map, it shows how it is shifting, the species is shifting from warmer area to colder area. So that will help the natural biologist to understand the better how and find out the ways how to manage the system. I think we have been educated a lot from your presentation. I hope this lecture will enrich or inspire our scientific community for working towards welfare of the mankind. Thank you very much once again.
Thank you. So now I shall take a few more minutes to present both the thanks. So it's an honor to conclude today's special talk on a mutation is a mutation by extending our heartfelt thanks to Professor Detlef Weigel for accepting our invitation and delivering this special talk. So your talk was so fascinating, and it will surely ignite our minds to think on the aspect you just presented. Also, this is a remarkable opportunity for all of us to listen and interact with you. Thank you very much, sir. And I hope for our stronger association with you in future also. We're grateful to have you. I take this opportunity to express our sincere gratitude to our respected director, Madam Dr. Padmini Swain, for presiding over today's program and for your constant support and guidance to make this program a success. I also thank Dr. Nayak, Head Crop Production Division, for delivering the remarks. I'm grateful to Dr. P.C. Roth, Principal Scientist and Chairman, Organizing Committee of Azadi Ka Amrit Mahotsav at NRRI, for planning and coordinating the activities. I am also thankful to Dr. Sudhamay Mandal, Principal Scientist, NRRI, and Member Secretary of this program, for meticulously planning and organizing the events. I thank all the head of divisions and regional stations, scientists, head of office, finance and accounts officers, administrative officers, and all other staffs of NRRI for attending today's special talk and providing necessary support. I extend my special thanks to all the directors, scientists, and researchers, students, and office staffs who are attending this event on virtual mode from across the country and from Germany and other countries as well. I thank all the staff of ADISL and committee members of Azadi Ka Amrit Mahotsav at NRRI for making all the arrangements and successfully organizing this special talk. So thank you all. So actually, that's all for today's program. So should we close here?
Yeah, let us close our session.
Okay, thank you, professors. Thanks once again.
Thank you. Thank you very much.
Thank you, Professor.
Bye-bye.
Thank you.
Thank you. Bye.